starting with the proper histoanatomy. We are going into the structure identification now, and we will be learning and proceeding to the rest of the structures that is going to come. The general part of this microanatomy is over. So, individual organ we are gonna project, and then we are going to identify how to identify them and how to separate between these particular similar like or likewise structures that we are seeing them. Now, one of the structures that we are going to learn today is the salivary gland, and the basic of the salivary gland we have begin with like with the glands that we have learned. Now, in your body we have a identifying pair of salivary gland. We call them as identified salivary gland, which is three pairs. We say parotid gland, sublingual and submandibular, three of these particular glands which are present in our body. And apart from that, we have some minor salivary gland. They are located elsewhere like cheeks, gums, soft palate, etc., etc., and rest of the tongue, cheeks, lips, etc., etc. These are the parts. Now, whenever you are going to study this particular salivary glands, we are going to mean that like the main or the prominent salivary glands. That means this particular parotids, sublingual, submandibular, we are trying to mean. Now, the basic framework of a salivary gland is that the functions of the salivary gland is to produce the saliva, which is basically having a functions of lubrications to moisture this particular food material. It needs some kind of hold some kind of degrading acids, which is degrading proteins or enzymes, which is going to split some of these particular substances that you are eating. And apart from that, it helps to create some kind of give some sort of antibody actions to combat some of these particular germs that is coming through the foods through some kind of substances. Now, these are the basic thing about this particular saliva or salivary glands, and this particular salivary glands, as we are seeing, three pairs of them are there. With this parotid gland is the largest of one, and then we have the submandibular and the sublingual. Sublingual is the tiniest one. Now, the middle one is the submandibular. But if you are talking about the utility and the functional capacity of this particular gland, so amongst these three, the submandibular gland is the word which is doing the most of the jobs. That means this submandibular gland is producing like 2/3 of the secretions of the saliva that is coming. Even though this parotid gland is the largest one, it is not doing that much of like the rest of the things, and this 30% or 2/3 is coming from this parotid gland and as well as this sublingual gland and minor glands. Now, if you go to these particular positions of this gland, submandible, that means it is lying below the mandible. We have the chin bone or chin bone over there, and this is lying below this mandible. That is why you call them as submandibular. Parotid gland is formed or present in face of the or on the side of the face, and sublingual is present inside this particular floor of the mouth, deep into the floor of the mouth, and it is opening and putting the secretions. So, all these glands, they are exocrine kind of glands, and that means they have a secretory material and they are putting or pouring this particular secretions, and they are throwing that in a tunnel system that we call them as the ducts, and that particular ducts opens into mouth cavity. Upon stimulus, this gland is producing this particular secretions and is pumped out and drained inside the mouth, and this is going on. And as it is required to have some kind of wearing by the nerves. So, we have some kind of nerves which is controlling these particular secretions, facilitating or slowing down the secretions likewise. This is how these particular things are working. Now, our part of these things, we are going to study this particular gland, how we are going to see this. So, whenever you are going to see this particular gland, we are going to make this particular small piece of them and see that what is the basic or unit of architecture. So, our aim of this class is to see that if you are seeing something there, we have to see that what is the tiniest unit or tiniest functional unit of this particular tissue that is existing out here. So, whenever you are seeing this particular gland, any of this particular major or prominent salivary gland, there are three things that is present in it. One is that we call them as secretory unit in anatomical terms, or this particular like a medical terms, we call them as acini. So, there are secretory hubs, that means they are producing the secretions, they are factories which are making the goods. Second thing is that we have this particular goods produced and they need to be transported out, that means it needs some ducts. So, it has a secretory unit, some houses is producing some kind of element, and that is kind of like taken out through the duct system and thrown to out. So, it has ducts and secretory unit. And to bind these particular things together, we have some kind of binding material, we call them as collagen tissues. So, that is separating between the collagen tissues with the collagen tissue is separating between these particular secretory units. So, between the secretory unit they have a partitions, they are made by the collagen tissues. Between this particular ducts and this particular acini, we have this collagen tissue that is separating these things out. So, giving a architectural support, that means it is giving a kind of structural unity support to this particular organ. So, this is basic architecture of this element. Other than this giving structural support to this particular gland or tissue itself, the collagen tissue which is present in it or the connective tissue which is present in it gives some kind of roots for that blood vessels, nerves to reach to it. So, it is paving the way how this particular blood vessels and the nerves reaching to this particular individual secretory unit or acini. This is how we are going to understand. So, it has a secretory unit, it has a output unit which is taking or carrying these things out, and separating unit which is known as connective tissue unit. Now, if you see this particular acini, the acini can be divided of three types that we have learned yesterday based on the function of the secretions that we are seeing. It may be pouring some kind of clear secretions, we call them as serous acini. If pouring some kind of thick secretion, we call them as mucus, and if it is two of them, we call them as mixed kind of thing. So, based on that, we can just like classify is a serous acini, mucus acini or mixed acini, alright, depending on. And this particular ducts which are present all inside to it, it is also be classified. I will be coming into it. So, ducts are very prominent in the salivary gland. They are very prominently branched with the ducts. So, they are prominence of this particular duct is a hallmark for this particular gland. This area have got very good amount or large ducts we are going to see. Now, ducts is that we have a duct, we call them as main duct or main salivary gland, that branches into smaller branches. We call them as in between the lobes it is going, right. So, we call them as lobar branches. So, main branch to the lobar branch, and then in between the lobe it is going, or we call them as interlobular branch, and if it is going inside the lobe we call them as intralobular branch. So, this is how we can classify it and name it. We are just like not going to emphasize this particular naming of this particular thing, but you have to understand how these things are put into it, so that we can remember this. So, we be like a main duct, lobar duct, interlobular duct or intralobular duct. What does it mean actually? So, you should be knowing that. Now, out of this particular element have got this particular duct which is present inside to this particular glands, just like this is the architecture of this, like schematic architecture. These units we call them as the acini, the factories. This is the connective tissues, this is kind of like the duct channel system which is carrying this particular secretions which is going there. So, this particular unit we can these 1, 2, 3 can be called as a lobe, for example. So, there may be another lobe and separated by a line which is known as the connective tissue. Am I clear what I am trying to say? So, we have this particular lobes which is present between them and separated by some partitions. So, in between this particular lobes we have a partition, that partition is created by the connective tissues. So, this is how these particular things are organized or placed out here. Now, this particular ducts which is present inside to it, they have the names how they are present. Now, the duct which is present within this particular lobes which is present, like this is how this salivary ducts can be divided into lobar branch, interlobular branch, intralobular branch, that means inside the lobe. Once inside the lobe, if you see any duct, we call them as striated duct or intercalated ducts. So, these two terms that you have to keep in mind regard to this particular ducts which is specific for these particular glands. What do you mean by this intralobe or this particular intralobular branch? That means suppose we have this particular thing is lobe. So, inside this lobe we have these small ducts. These ducts we give a special name to the salivary gland, we call them as striated glands or striated ducts or intercalated ducts. So, these two terms that you have to keep in mind. Once you show that in the histological picture, that will clarify what I am trying to say. So, this is how these particular salivary ducts can be classified, or this is the architecture that we are talking about. These are three sets of glands or have same architecture things. They have this particular secretory unit, we call them as acini. How the acini can be divided off? We understand how the acini are, what they are making actually, and ducts. Out of this particular thing is separated between them capsule, which is separating between the lobes and lobules. Now, how does these things looks like when if you are seeing them in a architecture? So, this is a schematic picture once again. So, you see that this particular element we can call them as a lobe. This is another lobe and separated by a septa like this. Got it? What I am trying to say? So, this is septed. So, this is lobe A, lobe B, lobe C, lobe D likewise. So, see that this particular lobes have got like a acini, which is secretory unit, and it also have some ducts. These are ducts and these are acini. Ducts have hole in it and you have to look close. This is another hole inside. So, this is the lobes which is having this particular ducts inside. So, we call these ducts which is present inside the lobe as intercalated ducts or striated ducts based on how they look like. If they have striations, we call them as striated ducts. If they are non-striated, we call them as intercalated ducts. So, this is how we are going to classify it. Clear? So, coming to individual glands or secretory unit beforehand. So, acini, what is that? This is the secretory unit formed by this particular epithelium cell resting on the basement membrane, and they are producing the secretions. How they are producing the secretions? We can classify as a serous, mucus and mixed varieties. That is the same. Serous cells, which is pyramidal to the block, protein rich, high in water content, and they are producing this particular protein rich chemical substances that is coming out. Mucous acini, that means they are producing this particular mucus rich substances. Mixed, they have this mucus capped by demilune, half of the moon or serous demilune, like that. So, these are the things that is present as a acini. Apart from that, we have some other cells which is present here. We call them as myoepithelial cells. That is a cells which is modified, which has a contractile power, which can squeeze this fluid out of this particular element. We call them as myoepithelial cells, which is pumping out the fluids which is present within this particular duct. So, these are the cells that is these are present between this particular ducts and glands. So, individually if you land up into the gland, parotid gland largest of all, and this particular parotid gland has a acini which is predominantly serous. So, we call them as serous gland as parotid gland. Okay. So, this particular serous gland has what component? Same thing, acini, ducts and connective tissue. What kind of acini you will be getting in here? Serous acini. Why serous? Because the secretions are fluidy in nature and it is high in protein content. That is why it is as simple as that. And it has got ducts. What kind of ducts? Lobar ducts, interlobular ducts, etc. So, the same thing. If you are going to see this particular duct which is present in between or inside the lobe, we call them as intercalated or striated ducts, right. So, this is what we have seen that beforehand. So, this lobe which is having kind of ducts inside, we call them as with a separate name, and this lobe which is present in between this particular this duct which is present in between these two lobes, we call them as interlobular ducts. Right, in between two lobes, lobe A, lobe B, we have duct, we call them as interlobular duct. So, got the concept how these things are there. So, these are the things we are going to see. So, do not dig deep into it, just understand this particular thing and just like let it flow how much as you can assume. Focus in the prime point that you need to understand. Not everything you are going to learn, not everything you are going to memorize out here. What is required from you? Just like learn the basic thing to identify and separate the things between them. Right. So, if you get this basic idea of a gland, how these things can be classified of with the simple architecture of this particular thing, that makes clear your mind. So, this basic thing you will be coming again and again. So, what we are going to see? Acini and ducts, and in between them we have partition, we call them as connective tissue, A, B, C, 3 elements. Acini are of 3 types, A, B, C. Ducts, out of here plenty of them. If they are present within this lobe, we are going to give them special attention, right, which is a hallmark to identify these things. Similar to that, from there we are moving to the another gland which is known as submandibular gland, which is making maximum things. Again same architecture, does not have anything special apart from this parotid gland. So, ABC, acini, duct and partitions, connective tissue. Acini, this is a serous predominant. Also have got serous predominance. We call them as serous, but it also have some mixed cells inside. So, some call them as mixed kind of parotid, mixed kind of salivary gland with a serous predominance. That means it is a mixed, but a serous predominance is there. If you call them as a serous gland, no harm to it, because it is mostly serous, 70% is serous. That means what do you mean by serous? Watery fluid, clear, that is it, simple thing, okay, do not make it complex. So, this is how it looks like, does not have much of difference between these two, right. So, only thing is that we are going to see white structures, right. This white structure is acini. White acini is because, right, these are dark acini because they have the ducts inside to it, because interlobular duct, intercalated or striated ducts, that is it. If they are outside, we call them as interlobular, in between lobe A, B, C. So, you have to find these septations, if you can find, if not, you understand this concept of separating it. Sublingual gland. This is another gland which is major variety. This gland is again the reverse thing, that means mucus predominant. So, we have this learned that other two things were serous predominant or serous kind of gland. This gland is a mucus secreting gland. That means you are going to see lot of white things. Why white? Because the mucus things are there. So, this architecture of the gland is going to be different. Yes, how they are going to be different? So, what you are going to see? More white patches. So, what things you are going to see? Which part is different out here? Acini part. So, acini part if you are going to see, it will be more white compared to this. And the ducts will be same or different? Same. And how about this particular connective tissue? Same. So, everything is same, more white. Okay. So, once again you summarize and go hit this microscope and see that yourself. What are you going to see? Salivary gland. What do we expect to see? Secretory unit and duct system separated by partitions, connective tissue A, B, C. First you identify which one is a secretory unit. If the secretory unit is separated from other, that makes this as a lobe. Within this lobe, we are going to see two elements. One thing is the acini which is producing the secretions. Either it will be dark stained or it will be light stained. That makes these things or a mix of things. You understand how to understand this. If you be able to see some kind of like apart from acini cells, anything else, that would be ducts. Duct can be identified by a slit present inside to it. The slit which you can see, it's okay. If you cannot see, it's fine. Sometimes it is not because of the error, you are not seeing that because you are not, that's not wrong. Because they may be under processed, they may not be visible. You need to dig deep into it. So, if you are going to see, if you are seeing some kind of ducts within this particular lobe, they may be are of two types, striated or intercalated, right. If you are going to see some duct outside this particular lobules, interlobular, etc., etc. Understand? Concept is clear. If you are going to see something like that doesn't look like this particular ducts like this, something else, it could be blood vessels, it could be fat tissues, it could be something else. That means connective tissue. Blood is a connective tissue, blood vessels is something. So, this is a connective tissue, same thing falls in here. So, we have this particular thing separated out, lobes, and between them we have a partitions, septas, septations, and everything is present in it. That is how we are going to understand these concepts. Okay? Any doubts? Okay. So, slowly go through it, A, B, C. Keep it simple as you can remember forever. Alright. 3 basic things and then we are done. Thank you.